Okay, let's try again. Start with bit.ly, cold chalk, going straight to a Google Drive public folder where one can read the annotation files. Optionally, the protein sequences in FASTA format. Notice that forgot to say this, the annotation files were quite big. It's 184 megabytes, which amounts to over 1 million entries. So back to the protein sequences. Again, you see that the ability to parse long text files by the web browser is practically unmatched by other environments. Now let's choose the DNA sequences, now close to 90 megabytes worth of DNA sequences. We should have as many DNA sequences as we just parsed protein sequences, but the parsing is going to take a little longer because now both sequences are going to be assembled into an entry that will be registered to the node reference in the annotation file. And we found a little over 400,000 such entries. Now we can look for a pattern, say cold chalk for instance. And in spite of the very large number of entries in each of the two lists, because the sequences were registered to the node annotations, we can very quickly find different types of entries, satisfy different types of constraints. Cold chalk, so all of them will have cold chalk somewhere. We found 6, there they are. Now let's look for something else, just to illustrate this multiple searching ability. For instance, let's pick this organism here. And let's see how many are there. How many annotations do we have for which we have sequence, nucleotide and both nucleotide and amino acid. And there they are. We found almost 1,000 of them. Now this is a good time to go look at the object where all this information is being kept. So we are in Chrome browser, Google's Chrome browser, which has a very good developer tools. Other browsers will also have them. Firefox is also pretty good. Internet Explorer, it's okay, but Chrome is really the best for this. So we can now go over the code. We can change the code if you have access to the folder where this JavaScript file is being kept. And by clicking on Escape, you also get the console. So the objects are here, how to parse protein sequences, how to parse DNA sequences. And let's look now at what these results look like. Starting with proteins. There we are, 431,000 entries, which is slightly more than the number of FASTA registrations we got, which means that for some of the annotations we have more than one sequence. So let's now just open one of them at random so you can see the amino acid sequences for each of these entries. Time to go to the FASTA file now. Again, cold chalk is our basic object. It's created right here in the beginning, cold chalk, and now all the methods are defined within this object. So FASTA. Now because I'm registering the FASTA entries here, I do expect a very large number of methods. So I'm going to use the native object and get all the property names within the FASTA object. And there we are, 427,000, matching this one here. Now let's look randomly inside. So these are just the indexes. So I'm going to pick randomly one of them. So this one. And let's go back to the native object, FASTA, and look for this entry. I lost something along the way, sorry about that. Let's do it again. Pick one of these, copy, and paste. Now we have an example of a registration for which there is only one sequence. You know there could be more than one. And for this node, 332784, there it is, we have a nucleotide sequence and a protein sequence. That's a funny artifact down here. This may deserve a second look. Why do we have the case changing halfway through the sequence? Let's pick another one to see if this is a general problem. Alright. Let's change the nodes. Again, only one entry. And there we are. We have the nodes information and the two sequences. Let's go to the end. I guess that was a isolated problem over there. Alright, let's see. Cool, so I suggest you now go over the object and find out if you understand the methods that were coded here. Remember, now that we have the results all stored in this code shock object, you can display the results any way you want, as a long table, as a tab delimited text file that you download and then say open in Excel, anything you want. So this is just the first pass. This is your homework. Have a look at this code, make sure you understand it, and we'll go from here. Alright, talk to you soon then. Bye bye.